This lecture is all about meiosis and genetic diversity. So to start, we're going to review two key terms that you should have learned in an earlier biology class, but if not, I'll go over it right now. So diploid is the normal state of human cells. We have two copies of every single chromosome, the maternal pair or the maternal set and the paternal set. So we get one copy from each parent. We have 23 pairs of these chromosomes, which gives us a total of 46. So how would we maintain that stable number of 46 chromosomes when we reproduce? We have to take one of our normal cells and essentially divide the amount of chromosomes in half to create a gamete, a sperm or an egg cell. That's what we call haploid. It contains half of the normal genetic information, or only one copy of each of these chromosomes. Then upon fertilization, where the sperm and the egg fuse, it's going to restore the diploid state. So you can see these chromosomes here, they only have one copy of each of them. This is a haploid cell, like your sperm cells or egg cells. And when these fuse together, you'll have one sperm with a haploid cell with half the number of chromosomes, and the egg is going to have the same exact state. When those fuse together, you'll end up with the two copies of every single chromosome, that diploid state. And you'll notice here that the colors of each of these chromosomes are slightly different. So it's a different shade of purple, a different shade of blue, a different shade of green. That's because while I'm still getting copies of every chromosome, they are not identical copies. Each parent is going to give a different copy of every single gene. There might be minute differences in them that are going to give you different traits depending on how they come together. So let's deep dive into meiosis now. Meiosis is the process of creating these gametes, these haploid sperm and eggs. And the eventual goal is to have sexual reproduction occur, where two different organisms are mixing their DNA to create a novel, unique third organism. The cell has to separate their homologous chromosomes so that the cells that we create are haploid, and we'll go into all of that. And there is a big difference between how this is done in human males and human females, and we'll talk about that at the end. So to get into meiosis and really understand what's going on, we have to look at the DNA and really see what's happening to the DNA during this process, because the whole goal is to get that DNA to a point where we can then create a new organism's DNA. Now, meiosis evolved out of mitosis. It was a process that came later in the evolutionary timescale. So the basic principles of mitosis are conserved. There's going to be a lot of repetition here, but one thing that does happen as a result is meiosis is going to happen after DNA replication. We are going to duplicate all of our chromosomes and then get into meiosis. So when we're looking at our chromosomes here, they originally start in this state when we're in G1. We have what are called homologous chromosomes in this particular instance here. The copy from parent number 1 and the copy from parent number 2. But these are identical chromosomes otherwise. That's why they're called homologous chromosomes. Homo means the same. So they're the same chromosome, like chromosome number 1. All the same genes are on there. I'm just looking at the two different versions of that chromosome that I have. Now, this is not the same thing as a sister chromatid. The sister chromatid refers to identical copies of that chromosome. So when we do DNA replication, each one of our homologous chromosomes is going to duplicate itself. That first copy is going to end up with two sister chromatids attached, and the second copy is going to end up with two sister chromatids attached. So this state, we often call it a tetrad, because we end up with 1, 2, 3, 4 unique chromatids, or almost unique chromatids in this case. The goal of the first part of meiosis is going to be to separate these tetrads. Then the goal of the second part of meiosis is going to be to separate the sister chromatids into their own cells. So we have a really interesting diagnostic tool at our disposal called a karyotype. This is where we wait for all of our chromosomes to condense, then we can take a quick picture of them, and from that picture we can sort them by their chromosome number. And this allows us to compare the chromosome pairs to make sure they're all looking very similar. And it allows us to do some quick diagnostics for diseases like trisomy 21. If we have an extra copy of the 21st chromosome, you have what's called Down syndrome. We can also see that the 23rd pair of your chromosomes is not labeled with a number. It's labeled X and Y. And this is because females inherit 2 copies of this X chromosome. Males inherit only 1 copy of the X and they get a Y chromosome. The whole purpose of that Y chromosome is to confer male characteristics on the organism. Now, meiosis, we're going to undergo 2 sets of cell divisions, and we're going to get 4 daughter cells as a result. These daughter cells are all going to be unique. They're going to have their own individual mix of the original organism's genome. So meiosis 1 is going to give us 1 division, and it's a reduction division. We're going to go from a diploid cell, a 2N cell, to a haploid cell, just N. Then meiosis 2 is going to take that haploid cell and divide it once again to create our 4 final haploid cells. Meiosis 2 is essentially identical to mitosis. Now, meiosis 1, the cell is going to begin an interphase, and it's going to go through essentially the same exact steps as mitosis. We break them down as prophase 1, prometaphase 1, metaphase 1, etc. You get the gist. It's just the stages of mitosis with a 1 attached to them. The focus of meiosis 1 is to take those tetrads, those homologous chromosomes, and separate them into their own cells. That is the key thing that makes the new cells haploid. I'm taking the 2 copies of each of my chromosomes and I'm separating them, so I only have 1 copy in each of my resulting cells, diploid to haploid. So at the end of meiosis 1, our original 46 chromosomes are going to be cut down to 23. Also within meiosis 1, there's a really cool step called crossing over that we're going to go over in a little bit. And the whole point of that is to mix things up even more within the cell, increase that genetic diversity. Now, here's just a picture of what's happening. We can see that the tetrads, these homologous chromosomes, are lining up, and they're going to be separated from each other in anaphase, and then we're going to reform that nucleus around them in telophase and split them into the 2 cells. Meiosis 2, as I said, is exactly the same as mitosis. We're going to take these 2 sister chromatids and we're going to separate them and split them into their own cells. So we're taking the identical copies of that particular chromosome and separating them into cells. The problem is, well, not really a problem, but the key thing to remember is these were haploid cells. I don't have the copy that I got from each of my parents in these cells. I'm only going to have 1 copy inside those cells. But when I do this division, the cells are going to remain haploid. Remember that sister chromatids bound together are considered 1 chromosome, and each chromatid, when it's separated, is still considered 1 chromosome. So we're not having the information again. We're keeping the amount of chromosomes the same. So we're going to start with 23 and we're going to end with 23. And the way we break down these steps, same way as mitosis, we just add a 2 at the end of their names. So this is prophase 2. If you're ever looking at an image and you're not sure, am I looking at mitosis? Am I looking at meiosis? Am I looking at meiosis 1 or meiosis 2? The way that I tend to remember it easiest is I'm going to end up with not one, but two cells going through the process in any image of mitosis or meiosis too. Another way to look at it is what's going on at the chromosome level. Here you can see I have sister chromatids, and those sister chromatids are being separated from each other. In meiosis 1, you're going to see that it's the tetrads that are being separated from each other, not the sister chromatids, and it's only going to be one cell. Here's another image just to put it all together for you. So we start out in prophase. In metaphase, we're going to line up all of our tetrads. We end up having crossing over occur really during prophase, not during metaphase, but you can see that this blue chromosome is no longer completely blue as it was in the beginning. We've kind of switched genetic information over, and we'll go over that in a few slides. The tetrads are separated, the homologous chromosomes are separated, and we end up with haploid cells. In prophase 2, we now line up our sister chromatids, not our homologous chromatids. We separate them and pull the separated chromatids to opposite sides of the cell. So let's get into the steps that the cell takes to increase its genetic diversity, well, the cell and the organism. So the first thing we're going to mention is crossing over. This is going to occur at the very beginning of meiosis, during prophase 1. Our homologous chromosomes are going to be so closely associated with each other that pieces of the chromosomes literally break off and jump over to the opposite chromosome. They switch places and then they reattach. So if you're looking at this image here, we can see that this red chromosome, it actually has a piece of the blue chromosome on top of it because they switch places, and the same thing happened on the blue. It has a piece of the red chromosome. So you can see how this is just going to increase the genetic diversity. I'm going to mix up my possible genetic traits even more. And if you look inside this image here, when we end up with our gametes, when we do crossing over, we end up with 4 unique gametes. If crossing over didn't occur, these two would be both completely blue. These two would be both completely red. And while that's still some uniqueness to the cell, it's not the same as what happens with crossing over. We're going to get incredibly unique and diverse options when we cross over, essentially infinite amounts of combinations. What essentially is happening is as the two chromosomes get closer together, they're going to have a synaptonemal complex form. Basically, it's proteins that draw them closer together. And when that happens, sometimes the legs cross over each other, as you can see here. And that crossing over point is called a chiasma. This can happen either once on a chromosome, where we just have one leg crossing over, or you can have a bivalent crossing over experience where it happens on multiple parts of the leg. And this is going to happen at least one time on every single chromosome in the cell that's undergoing meiosis. So you can see how if this is all occurring at random points, we're going to end up with vastly different cells. The next thing that increases genetic diversity is independent assortment. During metaphase 1, the homologous chromosomes line up randomly, meaning we don't have all of one parent's chromosomes on one side and all of the other parent's chromosomes on the other side. They don't line up nicely, chromosome number 1, then chromosome number 2. It's not like Noah's Ark. It's all kind of whoever gets to that point first is there. So it's more like possibility 2. We might have a mix of every single copy of the chromosome on either side. It's called independent assortment, this random alignment of all of our homologous chromosomes. And you can see that the difference it makes in the daughter cells that are produced. These ones will end up with all of, let's say, the maternal DNA inside, and this one with all the paternal DNA, whereas these have a nice mix of both the maternal and paternal DNA. Now, consider that we also do crossing over. You're looking at even more genetic diversity coming about when you combine the two. The final way that we increase our genetic diversity is through random fertilization. Whatever egg is released is done so at random. Whatever sperm is released is done so at random. The sperm that ends up fertilizing the egg is done so at random. Not only that, but there's sexual competition. Now, humans don't often have this because we're monogamous, fairly monogamous creatures, but in nature, you'll end up with one female copulating with multiple males in the same breeding season. And so you're going to have competition between males themselves, and then you even have competition at a sperm level where the sperm of different males are competing with each other. Now, when we're looking at just the human experience here from one mating experience, if you look at all of the sperm and egg possible combinations from that one mating experience, not including things like crossing over, not including things like independent assortment, the likelihood of one particular sperm and egg meeting to make you is 1 in over 70 trillion. So whenever you feel like you're just not special, or you're not feeling up to your normal self, just remember you are 1 in over 70 trillion possible humans that could have existed. So it makes you really unique and really special. So all of this just puts a real strong highlight on the value of sexual reproduction. Sexual reproduction increases our genetic variation in spades. When you're looking at mitosis, you're looking at asexual clones being produced. Every single daughter cell is going to be identical. Now that's great if I'm trying to make more copies of my cells to grow, but when I'm trying to reproduce and create offspring that are going to then pass on their genes, all of this genetic diversity is going to create a healthier, more robust species and organism. Because if my species has a wide variety of genetic possibilities, we are more likely to survive no matter what the environment throws at us. The environmental conditions can change drastically, and my genes have within them the ability to survive. And we'll talk about that a lot more when we get into evolution. So we're going to look at the two ways that meiosis happens differently in males versus females. In spermatogenesis, the process of making sperm, it's going to begin at puberty and can continue until the death of the male. So this happens a lot for a long time period. Millions of sperm are created every single day, but the overall process to make one individual sperm can take about 2 to 3 months. So it's not a daily thing that happens. There is continuous development though. So if you look at this example image here, these are early sperm that are developing, and then as they become more and more mature, they move towards the lumen or the space inside the testes, and once they're mature, they can then be released and potentially go and fertilize an egg. Now, every single cell that goes through spermatogenesis is going to create 4 mature sperm cells. So one cell can produce 4 mature sperm cells. And just something kind of fascinating, sperm are the smallest cells of the human body, and eggs are the largest. Now, females, it's significantly more complicated. Meiosis 1 and meiosis 2 happen at drastically different times. In the males, you're seeing them happening one right after the other. With females, meiosis 1 happens when you are an embryo. So while you are still in utero, meiosis 1 is occurring. But it only occurs up to about the end of metaphase 1. Then the eggs become kind of suspended, like they're being frozen for a trip across space or something like that. Then at puberty, the females will begin to ovulate or releasing those eggs on a fairly specific cycle. For humans, it just happens to be a monthly cycle. For other mammals, it happens differently. And at this point is when anaphase 1 is reactivated. And the little tiny egg cell will start to grow and develop until it is finally released. Now, that's just meiosis 1. We're not even going to finish meiosis 2 for most of the human female adult life. After meiosis 1 is completed, we create one secondary oocyte. It's kind of like an early egg, and then one polar body. These are really tiny byproducts. Like the second cell that's created is not really a cell. We spend most of our time and energy creating that one secondary oocyte. So females aren't going to create the 4 sperm cells per starting cell the way males do. We're only going to create one egg cell for every single cell that enters meiosis. Now, meiosis 2 begins right after meiosis 1 ends, as the egg is traveling down the fallopian tubes and into the uterus. But meiosis 2 does not complete unless it is fertilized by a sperm cell. So your eggs will never fully complete meiosis unless they are ready to become a new organism by being fertilized. When we do finish meiosis 2, we create that ovum, the egg itself, and another polar body. So, interesting thing is that females are essentially born with all the eggs that they will ever have, and most of them will never even fully go through meiosis. Only, 1 to maybe maximum 8 or 10, if you're one of those octomoms or something, are going to become eggs that have gone through all stages of meiosis. Humans also have menopause, where once we've exhausted our egg supply, we can no longer create more because it all happened when we were embryos. So we go through a point where we no longer release eggs anymore because there are none to be released, and we no longer are fertile. We can't have children anymore. Now, there's some really unique cases in nature. Organisms do not like to follow rules. Life likes to bend the rules and find unique ways of living life. So plants are a great example, particularly plants that have spores, like ferns. What they will do is they'll alternate between a haploid life cycle and a diploid life cycle. It's called alternation of generations. So the spores are the haploid life cycle, and the plant itself is the diploid life cycle. Now, bees have this really fascinating experience called haplodiploidy, where the actual gender of the organism is determined by whether or not they are haploid or diploid. And bees aren't the only ones that do this, but they're just the most easy to understand example in nature. So the queen and all females are diploid. The males, or the drones, they're going to be haploid. So males actually develop from unfertilized eggs. The queen will lay an egg, and that egg will just become a male drone. Now, when a drone does happen to fertilize an egg, that egg can become a worker bee, and all worker bees are then female, or that one worker bee who's a female can be fed that royal jelly, and that female will then become a new queen. So let's look at mitosis right up against meiosis and see how they compare. Mitosis, you only go through one division, whereas in meiosis, we go through the 2 divisions. We produce 2 cells in mitosis, but 4, theoretically, because in oogenesis we only produce the 1, in meiosis. Now, mitosis produces identical daughter cells. This is an asexual reproduction experience where we produce clones. There's no crossing over. Meiosis, every single cell that is created is genetically unique. Crossing over is going to occur in prophase 1, and we're going to increase genetic diversity. In mitosis, the ploidy stays the same. A 2N cell is going to remain a 2N cell, diploid to diploid. In meiosis, we see a reduction in that chromosome number. It's going to go from diploid to haploid. We do mitosis for asexual reproduction, growth, and repair, whereas with meiosis, we're doing it to produce gametes so that the organism can reproduce in a sexual way. And here is just a pictorial version of what we just talked about. Make sure that you know these. These types of questions comparing the two do come up on the AP test. And if you have any questions, let me know.